Hi, in this video we're going to explore how to import functional assay data and associate it with your antibodies for lead selection in Platforma. Here I have already a preset up project with a couple of samples coming from 3 rounds of enrichment. I annotated those antibodies with my XR clonotyping and did a bunch of other things, like for example clustering, enrichment, and antibody sequence liabilities prediction. So to import our functional assay data, we are going to use Immune Assay Data block. That's a universal block allowing to associate metadata with your antibody or TCR sequences. So let's dive into the settings. Well, first we need to select the data set, and here the next thing is what is the sequence in our data set that we're going to use to match the information of coming from functional assay with our NGS clonotypes. In the case of my project, I have a VHH data, which is sequence full length. So I'm going to use the full VDG region amino acid sequence as the sequence in NGS antibodies that I'm going to match with my immune assay data. And now the next thing I'm selecting the actual source of the assay data. And this may be different type of data. It may be a FASTA file with the information coded in the headers. It may be a TSV file, like in my case, I have a simple TSV file where I have a sequence column with the protein sequences of my very well-known good antibodies, and then whatever metadata columns you have. Like in my case, I have a column hit which just has a constant value of ty1. That's I know the information associated with my antibodies. But you may have a couple columns. You may have several additional columns with, for example, your binding information with other information. I have many of antibodies here, so I have just 7 of them, but your list may count thousands. It's not a problem for the block. So in our assay data, the column with the sequences is sequence column. And the only one column that I have in my table is hit column that I'm going to use to import and associate with the antibodies coming from NGS. The other important parameters are matching parameters. How we are going to match the functional assay sequences with our NGS antibodies. So there are 2 options for the alignment score, either Blossom matrix or exact match. I am using Blossom because it will take into the account also the biochemical properties of the proteins. And now the careful setting is threshold to consider to match this sequences with our antibodies. This score threshold is something that you need to consider carefully because it depends on what is the feature. Let's say if we match just by CDR3, then CDR3 is really short and the score threshold should be rather low. But since we are matching by the full VDG region with relatively long, then I'm going to increase the default to 95%. So if we have a matching score between our functional assay sequence from this column and NGS sequence of 95%, then it will be counted as match. So let's run this block. It will take a couple of minutes depending on the size of the data set. So once the analysis is complete, we have a list of our functional assay sequences here and the number of matched clones from our NGS data set. So for each of the functional or of the sequences that we have in our table, here we have the number of clonotypes that are matched. And yes, that's basically it. Now we can use this information downstream. The basic stuff like clonotype browser. In the clonotype browser, among of the other columns, we now have a hit column, which is coming from the functional assay data. And for every clonotype from our NGS data, we have this hit assignment. So this value is assigned only to those clonotypes that are matched with our functional assay data. Another example is clonotype space. Let's go to the clonotype space. And for example, here, let's use this hit column coming from our functional assay data. And, well, for example, highlight the antibodies on the whole space that are matching this functional clones. And as usual, let's do enrichment as a color and see whether we have some nice hits here matching our functional assay data. And we do have, which is nice. So that's it for today. Stay tuned for the next guides where we will explore how to automatically pick the best lead candidates, combining all of the steps above into a unified table with your validated leads.